So, I have a fairly ambitious title, Brain-Computer Interfaces Increase Whole Brain Signal to Noise. And rather than explaining all of that at once, I think I'll just start my talk and start with a movie. This is actually showing one of our first real-time fMRI experiments. And so specifically what you're seeing is a human subject moving a cursor using only fMRI images of their brain. And in these first experiments, we were doing fairly simple tasks. And so this subject in particular had a fiber optic button box and was just alternating between tapping with his left index finger and his right index finger. A very boring task. So to pull this off, we could have done a number of different things. The most trivial thing, which we did not do, would have been to just use the button presses themselves to move the cursor. But another thing that we could have done, knowing that this was a button press task, would have been to select appropriate regions of the brain, say motor cortex in the left and right hemisphere, and use the fluctuations of both signal in those ROIs as a control signal to update the position and orientation of this cursor. Well, we didn't do that either. So instead, what we're looking at is the output of a whole brain pattern classifier that has learned the fMRI intensity values that best discriminate between left finger tapping and right finger tapping. Well, how does that work? Well, if we consider this as just kind of representing a single time point of the brain, then we can essentially represent this volume as an n-dimensional vector, where n is the number of voxels in the brain that we're interested in. And if we then number these as, say, voxel 1 and voxel 2, we could represent at least two of these on this two-dimensional space. So this vector here is a single time point of the brain, and I'm just showing 2 dimensions of it, but this generalizes to 20,000 dimensions, say, if you're doing a standard sort of vanilla acquisition. So if these intensity values are changing over time as the fMRI task is progressing, then we can build up a data set such as these. And then the goal of classification then is to find a discriminant boundary, in this case a linear model, that best separates these two experimental conditions. So what we've done here is we've found a separating hyperplane that explains the data that we've already collected, but also allows us to make predictions on new data if we were to collect it. So if we were to collect a new data point that put us in this part of the plane, we'd say the person was doing the red task, whatever that is. And so the other thing that's important to point out is that this model then is very sensitive to the label structure that we chose. And I've kind of intentionally made it an ambiguous sort of distribution. We might have other structure that we haven't modeled because that wasn't part of our label structure. I guess the final thing to point out is that this model, this linear model, is defined by its perpendicular based on these coordinates, and that kind of gives you a rough sense of how we can obtain maps from this exercise as well. So for this particular case, if we needed to discriminate between the red and the blue data points, we would say that voxel 2 was more important in this case than voxel 1. And so roughly the weighting would proportionately scale to that importance as well. So how does this work in real time? Well, if we start with a block diagram that represents a prototypic fMRI experiment where you have a human subject in the scanner, you're generally asking them to do some sort of stimulus-guided task, and then you're collecting a sequence of images of the brain over time. What we've done is we've actually hijacked this imagery construction system of the fMRI process and given it 2 additional capabilities. One is computer I/O capabilities, so that the stimulus presentation computer can talk with the imagery construction system. This is important for giving us the label structure for our data. And the second is compiling into the imagery construction code the ability to do machine learning. So in this case, using support vector machines during data acquisition. So we think about this configuration as running the scanner in training mode, where we're not training the subject that's in the scanner, but we're collecting data so that we can train a supervised learning model, so that we can then relate these images to what's happening experimentally. And then once we have that model, then we can decode new images as they're being collected and potentially use that then for neural feedback. So in other words, as we're collecting data, we're trying to decode what the sensory, behavioral, or otherwise inner state of the person is as each image time point is being collected. So how did this work for our button press experiment? Well, these images show the raw output of the classifier, which we were thresholding to update the position and orientation of the arrow. The dark, bold kind of time series illustrates just the very simple, repetitive left-right alteration between left tapping and right tapping. And what you can see is that even in this first bare bones implementation, this is a pretty robust process. Well, I still haven't explained why I went through all the work of building a pattern classifier rather than just tracking, say, motor areas of the brain. Well, I think that there are a number of advantages to this approach that we're using, but in my mind, one of the most important advantages is experimental flexibility. And so, without changing any of the workflow on our side, from the control room point of view, subjects have been able to control this exact same experimental interface by doing a variety of different tasks, such as thinking sad or happy thoughts, thinking in Mandarin versus English, or even doing imagined motor tasks rather than affected motoric acts. And so with just actually a day or so of extra work, you can build different types of interfaces where you might have a very task-relevant paradigm where you're showing smokers cigarette cues and asking them to modulate their craving to cigarettes. Or you could build generic interfaces that have very simple ability to give subjects feedback and present simple instructions. Yeah.
Sorry, if you're running a classifier and not actually targeting ROI, how do you have any assurance that the subjects are actually doing those manipulations? So in other words, couldn't they covertly be saying left, right, instead inside their head?
Yeah, yeah. Well, the burden of interpretation is on you. So one thing that you get is you get a map that relates to what's going on. And so actually I can give you a pretty simple example of that. And this might be a theme through the rest of the talk. So ping me again, or I'll try to keep the point in mind and kind of evolve the point as I go. But one of our first kind of pilot trials with this experiment, we had a bunch of smoking cues, and we wanted to actually give the subjects, we've now tightened down the instructions quite a bit, but we wanted to actually see what subjects would do. And so when we were asking them to crave, the first subject did great in terms of being able to move this slider bar between crave and don't crave. But a lot of these images are faces, and what we ended up seeing in our map between crave and don't crave looked like a lot of fusiform. When we went back and we asked the subject what they were doing, and we actually have eye tracking data with this as well, but for the crave they were watching the stimulus, for the don't crave they were actually avoiding it and watching the slider bar. And so, you know, we ended up with something that maybe was partially crave, don't crave, but was also partially faces, non-faces. As part of our task. So it's a very important point. You know, one thing, you need to interpret the data that you have, and for almost everything I've done to date, there's at least some thread of a literature that can kind of give you some plausibility. You can build group maps of these models, for example, and see how they relate to previous literature. But the exciting thing is, to do covert tasks. It's not so exciting to do button presses left and right, but you get a great behavioral measure if you do that. Okay, so these may look very different from marching a cursor across the screen, but really what's under the hood for all of these is virtually identical. So the engine under this is exactly the same. And so part of what that means is that I've been free writing on my own work now for a few years, which is somewhat nice. We're not limited to linear discriminant boundaries. We can very easily incorporate nonlinear boundaries by using kernels. We haven't done that very much for technical reasons, but not because it's a technical limitation. We can also generalize beyond, I apologize, my phone somehow is now connected to the computer because I use it as my thumb drive. Okay. So we can have multiple categories of stimuli. We're not limited to just two classifiers. And also if we have an experiment that's more parametrically designed, such as if we wanted to quantify level of craving of cigarettes or rate of finger tapping, then we can cast that as a regression problem instead of a classification problem as well. My lab for several years now has had a plugin that's available through AFNI. So if you use AFNI, you have this plugin. It does. It started off as a tool for doing offline analysis for support vector machine analysis of fMRI data. It still does that, but it also can operate in real time too. And so it's very well integrated with AFNI's real-time fMRI capabilities. And so actually this block diagram that I showed where we were essentially doing all of our processing on the MRIR has kind of evolved over time as well. And so actually we've shifted more and more of the work off of the image reconstruction box and onto a separate AFNI box. It's a little bit more modular. The MRIRs give you a lot of cool capabilities, but most of that we weren't utilizing. And the other thing to note is that when we were watching that arrow marching around, that was all that we saw. We could see successful arrow movement or non-successful arrow movement, but when we integrate things into AFNI, then we kind of have a real-time way to browse what's going on. So we have some in-flight viewing of what's happening in the brain. We can watch motion and correct for motion, and I was getting to the point where I was thinking about implementing all of these different pre-processing things that are already well made and well tested in AFNI, and it just seemed to make more and more sense. I threw in some basic benchmarks, and now as this slide is up, I'm realizing if hard-pressed, I'm not quite sure which system this has come from, but classification on the MRIR is extremely fast, because once you've trained the support vector machine, actually testing it is essentially a dot product of your new image with the model that you've already calculated. So all of this computationally works out quite well. The computationally intensive part is training that support vector machine, but once you have it, testing can happen extremely quickly, much faster than the actual acquisition that's required for your EPI setup. And so we have huge safety factors in terms of actually doing the classifier. And adding AFNI to the setup slowed things down quite a bit, but we still have a huge safety factor. So really, network I/O is the rate limiting step. But if we are collecting something like 30 slices normally, we could do 20 times this before we start bogging down the system. So we kind of artificially have done this to kind of see what our limits are. in terms of network capabilities. This next slide is really, I know, too busy to read, but if you were to come and we were to kind of do a dog and pony show for you to just kind of show you how this works, the main point of this left column is just to say that we do a few initial scans and we can really get the system up and running on an individual subject in a matter of 10 minutes with about 6 minutes of training data and 4 or 5 minutes for NPAIRS to start things off. Yeah?
How robust is that training run? So if you do 2 training runs, does it radically improve your classification, or does it matter?
I'll show one slide where it doesn't matter a lot. If we came and we were doing a dog and pony show, we would probably do the button press task, which is very robust. So 6 minutes always works for any subject that we've ever tried it with, but it can vary based on tasks. The right part of the slide also is no longer completely accurate in terms of how we're setting things up, and now I've kind of left it in as a public service announcement that once you start connecting lots of computers together, you might not know when to stop, and so you should just kind of realize that before you get going on it. So kind of the core of the talk, I'm going to go into a little bit more detail on each of these 3 experiments. And each of these could be a talk on their own, because from these interfaces and tasks, we end up getting support vector machine maps that relate to what the subject was doing or was supposed to be doing. But fortunately, I'm not going to give 3 talks today, fortunately for me and for you. But rather, I'm going to just kind of zoom in on some interesting things that we found in common with all of these 3 experiments. So we've really gone through, there's not a lot of mystery for this left versus right finger tapping. But when we were starting to develop this system, we were very keenly aware of the fact that bold fMRI has some major limitations. And one of the biggest ones, of course, is the hemodynamic delay. So I'm happy to talk more about this after the talk or during the talk. But that's kind of something that we were really interested in characterizing. And we were also interested in building tools for assessing these different tasks, for figuring out how much training data we might need for a given task as well. But really the point of this talk, which goes back to this idea of brain-computer interfaces increasing signal to noise, really starts off as a footnote in that paper. And what I was saying at the time was we have initial data suggesting that these permutations, and I'll explain what this means, may not be completely identical. Specifically, we see a slight tendency for higher prediction accuracy in run 4, regardless of training run compared to runs 1 through 3. So just as kind of a background for that motor test, I guess to make it really boring, I don't know exactly what I was thinking, but we did 3 runs where the subject was just doing the left versus right tapping and getting no feedback at all. And then in the 4th run, they were actually told that they were controlling the cursor and the cursor was moving around, and they were able to do this. And the figure that I wasn't courageous enough to put in the paper was this one. It turned out that when we were testing the feedback run, we tended to get higher classification accuracies than when we were testing on other runs. So we took the data back to the lab and did cross-validation. So we would train on one run and test on another and look at our classification accuracy. And these red arrows are what the real-time fMRI run represented. So training on run 3 and testing on run 4. But even for this one, where that's not necessarily at the high end of the distribution, the other runs were also related to testing that 4th run. So the higher prediction accuracies seem to come from classifying the run where the subject was actually engaged in moving the cursor. So that was something that I was interested in. We had a lot of other results, and for this we only had an N of 4, and so it just kind of was put on the back burner. Then a few years ago, we actually got fairly involved in a speech project, and this was while I was at Baylor College of Medicine, and collaborating with a speech therapist there, Monica McHenry. And one of the things that we were interested in looking at was rate of speech, which is a very important diagnostic for a lot of speech issues. So diadochokinetic rate is characterized for things like speech dysarthria. And so we had subjects do an experiment where they were supposed to covertly count. And I'll kind of play Monica McHenry's recording of the kind of behavioral target that they were supposed to try to get to.
1, 2, 3, 4.
So that's very kind of slow and smooth, and as opposed to this. Oops. See if we can get this other one to play. Oh, it's too bad because.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11.
So very forced and fast speech. And so what they did was they were supposed to practice this aloud and then get to the point where they could do it covertly. Well, this is a covert task again, and so we can't necessarily be sure that they were doing it, but we did have a couple of tricks. One thing is we randomized the time that we were asking them to do this, and so we would stop occasionally and have catch trials and say, what number did you get to? And we had a pretty good idea what range they should be in if they were actually complying with the task. Well, we were interested in doing this task, but we really had no idea how possible it would be to adapt to our real-time system. And so the main way that we characterize that is through classification accuracy. So if we're asking them to count slowly, can we actually predict that they're counting slowly, and to what degree can we do that? So the real-time setup is such that we do a training run where we're asking them to alternate between covert slow counting and covert fast counting. And what we get is a support vector machine model of them doing that. And
6-minute training too?
What's that?
Was it a 6-minute training?
This was about 10 minutes actually. And so we wanted to report what our percent accuracy was, and kind of a standard thing to do is cross-validation. And so a very simple kind of 2-run based cross-validation would be to take the data back to the lab and train and test in the reverse temporal order. And if things are exchangeable, then we would have 2 estimates of accuracy that we could average together and report either success to the rest of the world or abysmal failure, and move on with our lives. So what happened? Well, actually, things did work quite well when we were doing the real-time fMRI setting. And then when we went back to the lab, things also worked well, but we again got this systematic decrease in classification accuracy. For these same subjects, we actually also had them do that motor task, this left versus right tapping. And so we got a similar story there as well, where the real-time run was classified at a higher level than when we do things in the reverse temporal order. So I was pretty excited about this, but I felt that we needed to actually, we at least had enough information to try to go after this earnestly. And so we designed an experiment to do this. So up to now, everything I've talked about has had a training run where the subject's not controlling an interface, and then a feedback run where they are controlling an interface. And so there's a temporal confound there in that. Is that related or?
Finish your sentence and then I'll.
Okay, so one thing that we were worried about was something that was uncontrolled in terms of time order. So scanner drift, subject fatigue, that sort of thing. So we wanted to try to decouple time from this idea of feedback. Yeah.
I was going to ask what else was different about the visual display for the subject. So they weren't driving it. Was the visual presentation different nuance?
So in that first pilot, the display was just static. So now in this one, they saw something like this. So they were actually seeing a roughly matched display as well. Yeah, so that was another thing that we controlled this time too. So we didn't want that to be kind of a confound as well. It's a great point, and I would have forgotten to make it. But yeah, so they were told that they truly were not controlling the needle in some of the runs. However, the needle was moving. You could tell that they weren't controlling it. It was smoother motion. So we didn't do like really fine grain control and do like, but anyway, they were getting roughly the same amount of needle movement in both conditions. And then in some cases, they were told that they were controlling the needle, and they actually were. And so we randomized these in time, and we were able to replicate this classification asymmetry. But because we had these 4 runs to play with, we could look at some other things. If we had done a conventional fMRI experiment with no feedback at all in the mix, then this would have been the result that we would have gotten from a MVPA sort of analysis. And then one thing that we were excited about, because we had never tried this before, was actually training when the subject was controlling this interface and testing as well. And so we actually got a very dramatic boost in classification accuracy that I don't know if I would have predicted from the onset of the experiment. But I was very excited to see it. So where does this classification accuracy boost come from? That was kind of on my mind. And when I was looking at these support vector machine models, so for display niceties, I've thresholded these, but these are non-sparse models. These are continuous throughout the entire brain. But squinting at what was different between fast versus slow classification with no feedback and fast versus slow classification with feedback, these are not identical, but it's actually quite challenging to say that these are statistically different from each other. I know that because I tried, and you know, nothing that we tried actually was able to reject the null that these are the same.
Great. Can I ask a question?
Yeah.
So when you say you're trying to reject a null, that's like a mass univariate test?
Well, we did a mass univariate test. We also thought about these as distribution, so we did distributional tests because these.
Like a map level test?
Yeah, like a map level, exactly. Like now I'm blanking on the name. Kolmogorov-Smirnov test. So we did. Of course, we did kind of a second level type of analysis, GLM based, but we also did a Kolmogorov-Smirnov test.
You can't treat them as independent anymore, right? I mean, they're like the SVM weights are a linear recipe that says take this and multiply that by 2 and subtract this by 3. So it's an intrinsically multivariate map.
Well, you can.
Even looking at weights is somewhat misleading, right? They're not. A bigger weight doesn't mean more activation in the spot is more predictive of the +1 class versus the -1 class. Maybe if this weight is, if this area is higher than that area and less than this area.
Yeah.
Right? So it's really difficult to interpret maps as univariate maps.
That's right. Yeah, that's exactly right. And so, what we quickly fell back to was a method that was actually proposed for exactly this problem by my thesis co-advisor, Stephen Strother. And so what he proposed was a very simple idea of reproducibility for these maps. So if you have a pattern where, say, this voxel is high and this voxel is high, then it's going to obviously plot in this corner of the world, and similarly low would be here. So you can quantify this quantify the similarity of patterns. Say if you're training on run 1 versus run 2, or a run with feedback or a run without feedback, you can quantify this through things like mutual information or Pearson's correlation coefficient. And further, what was described is that if you do correlation, then you end up with a correlation matrix. Obviously, one pattern with itself is perfectly correlated, and then, say, a feedback run with a no feedback run would have some correlation value. you can decompose this correlation matrix using singular value decomposition. And the first eigenvector gives you one direction in the space, which we'll call the signal direction. And then the second one gives you a direction that's orthogonal to the first. And it turns out that the eigenvalues related to these eigenvectors are related, I'm using a lot of relateds, to the variance that along these two directions. And so you can do some simple algebra and come up with an estimate for the signal to noise, the spatial signal to noise of these models. And so we were looking at things like spatial reproducibility and classification accuracy to come up with kind of an analysis framework for evaluating these multivariate models. So what we get then are two performance metrics, prediction accuracy and reproducibility, from this kind of cross-validation or resampling framework. And so when I was a graduate student with Stephen Strother, we were building these sort of prediction accuracy versus reproducibility curves. So prediction accuracy, reproducibility curves, and then we were looking at the modeling complexity or how flexible our models were. I'm not going to go through a lot of the details in this plot, but the reason that I put it up was just to simply point out something that's a little bit non-intuitive in that you can have actually fairly high reproducibility with fairly low prediction accuracy and vice versa. So you can have, it's possible to have higher classification accuracy even if your model is spatially less reproducible. However, for our data, what we found when we did the difference between classification accuracy in the feedback condition versus the no feedback condition, and did the same subtraction for the SNR estimates that we were getting, so our spatial correlation, then we got this pretty robust linear fit between the two. And when we looked at what was happening, just collapsing over whether or not it was fast speech or slow speech, but just looking at speech with or without feedback, then what we ended up seeing was this frontal parietal system with Brodmann area 40, and then this frontal system starting up at frontal eye fields and following this whole swath down, and actually wrapping around to anterior cingulate, as well as a bunch of areas that kind of decreased in signal as well. And so where we are right now is a fairly complete picture of what's going on. We were motivated by trying to pin down what was happening with these classification effects that seemed to depend on whether or not you were classifying a run where the subject was getting feedback or not. We think that we have a handle on that based on the change in SNR globally that seems to be happening in the brain. And at the same time as these changes in SNR are happening, we're also getting this frontal parietal system and anterior insula, which, you know, yeah.
Sorry, I'm confused by the maps. Is that univariate analysis or is that weights on your classifier?
It's actually both. This one is a univariate analysis, but you can build a classifier of just speech versus no speech, and then if you essentially do like a second level analysis on those for feedback versus no feedback, then you get that as well.
Basically the same.
But because of some of the complications of, you know, multivariate versus univariate, this is a univariate kind of, if you want to call it traditional second level analysis, where we did speech contrasts at the first level and feedback versus no feedback in the second level.
So no classifier here, this is just standard fMRI.
This is standard fMRI. But you see the same thing if you kind of do the analogous process with the classifier. So, you know, this network has been, you know, kind of named or labeled an attention network, and anterior insula has, at least in a few papers, has been reported to potentially engage or disengage attention and also maybe turn off task irrelevant systems. Whether or not all that is true, you know, I'm not in a strong position to say it, but I'm more comfortable calling this frontal parietal rather than attention. But it seems that it seems plausible that these three are interconnected to each other. And so it kind of makes sense, just kind of stepping back for a moment and thinking about an fMRI experiment. You're basically in a tube, and someone's asking you to do boring stuff, like tap with your right index finger or tap with your left index finger. And even if you're a good subject and doing the task, you can be doing lots of other things at the same time. You can be thinking about your boyfriend or your girlfriend or how you're going to pay the rent or whether you turn off the burner this morning. So when you're giving someone feedback, though, you're perhaps engaging them a little bit more in the actual task, and it's a little bit more lifelike because what you're doing, either in your head or through very simple behavioral acts, now actually have some ramifications in the outside world. And so that's kind of where we are in terms of our maturity of thinking about this sort of thing. And so just kind of quickly, I'll talk a little bit about the smoker task, but really just to support the observations that we got in the speech task. So this is in collaboration with a colleague, Pearl Chu, who's a co-PI on this NIDA project that we've been working on together. And we've been showing smokers smoking cues and asking them to modulate their craving to these cues. And we've been quantifying, you know, again, we want to kind of as our step 0 be able to report to the rest of the world whether or not smokers can do this task. Can they move a slider bar to the crave direction or the no crave direction? And I apologize, I haven't updated this slide in a while. We have a lot more data and a lot more subjects. This is not intended to be any sort of political statement about Democrats and Republican smokers. But kind of the main point is that we've been doing this task and kind of going through the same motions. And some of the conclusions about this is that it does indeed seem that we can predict craving versus non-craving, and that these participants can control a neurofeedback stimulus derived from these craving-induced states. We've brought people back on multiple sessions, and so if you tend to be able to move the slider bar around well the first time you come in, we also find that people do well the second time. If they don't do well the first time, they usually don't do well the second time either. So we're not getting for free any sort of boost in performance by having people do this more than once. But at the same time, we are showing that what we're getting is pretty reproducible within subject. But kind of for the theme of this talk, we're getting this, again, classification accuracy asymmetry, and when we collapse against crave, don't crave, and look at feedback, no feedback, then we're getting this frontal parietal system as well. And if we zoom in, say, on Brahman area 40, and we do a median split of low accuracy subjects, the ones that aren't really controlling the cursor that well, versus high accuracy subjects, then we actually see that for the low accuracy subjects, we're not getting much change in this area in terms of percent signal change with or without feedback. But for the high accuracy subjects, we are getting this change. So now we're starting to try to, you know, we're getting some confidence that this sort of frontal parietal system is involved in this classification accuracy signal to noise story, maybe fleshing out. And so we're thinking about ways in which we want to use this. And so one question that we're asking is, can we use the frontal parietal network to evaluate feedback interfaces? So because we're interested in covert sorts of tasks, we don't really know if subjects are performing. We don't really have a great behavioral performance proxy all the time, and even self-report may not be that reliable. We want to see if we can actually maybe use frontal parietal as a signal that we could maybe evaluate different feedback options. So again, not going through a lot of the detail, we've been doing some more fMRI experiments. These are not real-time fMRI experiments, but very simple, just motor feedback, simple finger tapping sorts of tasks, to see if any of this holds up. And so, just to kind of, since the slide is up, just to kind of explain it a little bit, we've been using a visual metronome, and subjects have been just passively watching the flashing of this symbol, just kind of tap, tap, tap. Or they've been actually trying to tap along with this, but with no feedback, or actually getting some degree of feedback. So they'll have to follow the plus symbol, and when they hit a button, the circle will come on. And so based on that, you can see whether or not you're tapping at the same time as you're supposed to be. And we can do this with perhaps more information, and that was something we wanted to test, where we could actually change then the color from red to white based on whether or not you're in phase with the tapping as well. And kind of a variation of the theme, we have people just watch this flashing and then essentially keep an internal representation of this rate, and then they were asked to produce the tapping themselves, either with no feedback, with very kind of fuzzy feedback. You're just told that you're somewhere within the range of these bars, or with precise feedback. This is exactly the rate that you're tapping based on where you're supposed to be, and then giving them a chance to review. And so again, we see this frontal-parietal percent signal change differences between passive and no feedback. And you know, right now this is a group of something like 15 subjects. So you know, based on and these are standard errors. So you know, right now we don't have significant difference between feedback 1 or feedback 2, but we can certainly pull out the fact that feedback seems to be better based on this metric than no feedback, for example. And this delta prediction accuracy, delta SNR, also seems to be holding up. So we're fairly excited about this kind of stream of research. And then one of the other things that we've been thinking a lot about is this idea of, yeah, we're asking subjects to do things that are in a tube. And so covert tasks or imagery tasks are unrestricted, but you can't have subjects going in the scanner and dancing and other sorts of, you know, very active sorts of things. It's just kind of a limitation of our current measurement capabilities. But one thing that we can measure fairly well in the scanner that's not inhibited is speech. So going back to this idea of speech, we were interested in doing it with fMRI, and then we have started collaborating with Brad Sutton at Urbana-Champaign, who's developed a bunch of snazzy sequences to dynamically measure speech. And so this is Brad's lips and soft palate and gigantic tongue, and it turns out we all have gigantic, ugly tongues, but the idea could be someday that we might be able to evaluate giving people neurofeedback versus actual structural feedback based on speech production. And so we can apply the same kind of analysis tools for these data as we could for the fMRI data. And so one of the very first things we started asking is, you know, since we have these fMRI results, can we look at fast versus slow counting for these dynamic speech images? But when we started, and it turns out that we could, and this is a fairly good accuracy actually, because when you consider a lot of these frames are really dead in both, you know, there are gaps in, there are no speech frames in both of these series versus fast and slow. So we thought that we were doing pretty well, but when I started looking at the signal in the areas that the support vector machine told us were important, such as the lips, the tongue, soft palate, then I started to get really excited because looking at these time series, it looked like there was a lot of reproducing temporal structure here. And so we essentially jumped from this kind of classification of fast versus slow speech and decided to see, well, can we actually mouth read or decode what the person was actually saying in the scanner. And so the basic idea was that we could build spatial temporal models where we could play essentially the movie to a classifier, and with it being naive to the audio that went along with it, make a guess about what was being said. So it turned out that it worked most of the time. Pretty.
Describe the features.
I think like these, or
The inputs to the SVM.
yeah. Well, sorry, like this. What's in here? What's in this? Yeah. So this is actually a multi-class classifier, and so the way that we've done that is to use a directed acyclic graph. So what we've done is we've actually built classifiers of 1 versus 4, 1 versus 3, 1 versus 2. And so what's played into this is rather than just having one frame be the vector, what we're doing is we're actually having a clip of frames that we're stacking together. So then the components of our vector are then kind of spatial and temporal. And so then essentially a decision is made as you traverse this graph. And so then another tricky part of this is that we have these spatial temporal SVM models, and this is where I try to break PowerPoint. And on my air, it really does break PowerPoint. And I have no idea what any of this means. But what we're doing now is collecting new data that will hopefully make these spatial temporal patterns easier to interpret. So we're doing things like collecting syllables like pa, ta, ka, which we would expect to go from the front of the mouth, pa, to the middle, ta, to the back, ka. And so we've collected some data like that. We don't have the answer yet. I have a graduate student who's analyzing these data. But the idea is that we can look at these models and hopefully going front to back, both spatially and temporally, so that we can start to interpret these. Yeah.
When you asked the patient to do it covertly, did you notice any resembling toe movements in attention?
We didn't. We did not have this sequence running when they were doing it. They were instructed very carefully in terms of practice it overtly, then go covertly. The experimenter was observing the subjects as they were doing it covertly, and then we were also collecting physiology on the subjects. So we have respiration, but we do not have measures of what was happening in their mouth when they were doing it covertly. The biggest thing I was worried about for the fMRI was people tensing up and changing their breathing when they were doing the fast force counting. When I try it, I tend to do that.
Do you know how many support vectors you end up with? Is it actually sparse?
It's not sparse. It's
So
not
they're all
sparse.
support vectors.
Yeah. And so then the goal is to eventually, and this is the one movie that doesn't play, but it's not a big deal. What you would see if it were playing would be the mouth moving, and then a sweep of this kind of dynamic EPI interleaved with this. And so this again is a sequence developed by Brad Sutton at Urbana-Champaign. So to just kind of conclude the talk, whole brain classification enables experimental flexibility. So I've been able to kind of free ride on this and do multiple tasks. But for every new task and every feedback interface, our current thinking is that it's important to establish a positive control. So we need to show that smokers, for example, can move this crave versus don't crave bar and quantify that in terms of classification accuracy before we jump into something like a rehabilitation or therapy program. Because if we don't have even this basic capability, then when things aren't working in the future, we have really nothing to fall back on. So this positive control idea, I think, is very important. So we need to show the classification of the states that you're trying to classify as possible, and that subjects can then use this to control the interface. This NPAIRS framework is a very useful framework for data analysis within this setting. And specifically what we found by using NPAIRS is that controlling an interface increases spatial signal to noise. So, and this seems to lead to improved classification accuracy. So we're hopeful that this sort of finding could lead to new real-time fMRI approaches, and that this frontal parietal signal might give us kind of a way to look at covert tasks in a quantitative way in terms of how to optimize real-time fMRI interfaces. You know, keeping all of this in mind, we also have to keep in mind the fact that fMRI tasks are generally tailored to study specific components of behavior, so they're often pretty reduced experimentally. So you're just, you know, tapping on a button box might be over-reduced because it doesn't necessarily have any ramifications. You're tapping either successfully or not, but if you're not moving a cursor or controlling something, it may be less engaging. But the idea is that if we can get better measures of behavior as we're actually collecting the fMRI data, then hopefully that can move us in a direction of better brain behavior relationship understanding. And so then just to kind of conclude, the speech project was done in collaboration with Monica McHenry, with a lot of technical input from Will Curtis, Jason White, Jonathan Lisinski, experimental help from Dorina Papagiorgio. This motor rate stuff that I showed at the end was done by a very talented Rice undergrad who spent a year in my lab, Jeff Zimla, as well as technical help from Jonathan Lisinski. A big kind of commonality here is Jonathan Lisinski, who's really crucial to the operations of my lab. He's also helped out with the speech work with Brad Sutton, and then the Smoker project with Pearl Chu and Brooks Kinkasas. So I'd like to thank my past and present funding, and thank all of you guys for bearing with me for this talk.